cell cycle and mitosis. In this video, we're going to be taking notes together and showing some pictures about the cell cycle and mitosis, which is really kind of like looking at the life, I'm putting that in quotation marks, the life of a cell involving cell growth and cell division. I give you a little bit of a visual, and I'll be coming back to this here in a little bit. This is kind of what we're talking about. So think of it as a cycle with the idea that the beginning of the cycle would be here, and a cell would start to grow and go through normal cellular processes and get to a point where it has decided to divide and go through a process that you probably already know as mitosis, small little steps called cytokinesis, where we end then with two identical daughter cells. So this is what we're going to talk about today in the video. And like I said, I'll come back to this picture every once in a while so that we can take a look at it. So let's first start by just identifying very quickly that this process is slightly different depending on what kind of cell you're looking at. In a prokaryote, this is called binary fission. And you're going to learn about binary fission in an activity that we're going to do in class where you read about some different processes of mitosis, some organisms that do some different things with mitosis. So you'll read a little bit more about binary fission. And so in this video, we're not going to really talk about binary fission all that much. But I did want you to know that it's then different for a eukaryote. In a eukaryote, this concept of cell cycle and mitosis, you probably know better as interphase and mitosis, with mitosis consisting of prophase, metaphase, anaphase, and telophase. So when we put the interphase and the mitosis together, that whole concept then is the cell cycle. And just as a heads up, sometimes people even really consider interphase part of mitosis, but mitosis is the actual dividing of the cell, and so as a result, it's slightly different. So let's talk about something really important, and I'm going to kind of box this off because this is an important thing to know, is what is the purpose of mitosis? Why do organisms do it? So there's a couple of things. The first thing that they do is they are trying to replace dead cells. A great example of this is your skin. Every day we slough off thousands and thousands of skin cells, and obviously we need to replace those, and so we do that through this process called mitosis. A second thing is possibly to repair some damaged cells. And this is really a concept like small paper cuts or abrasions. Maybe the cells are dead or they need some repair, and so we're going to rely on mitosis to kind of heal those wounds. One of the big concepts that we use is we use mitosis to grow, to get bigger. We're increasing the number of cells in our body over a time. And then for some organisms, they use mitosis to reproduce. And that concept is called asexual reproduction because it only requires one parent organism to actually make an offspring. And again, when you learn about binary fission, you learn in more detail about asexual reproduction and some examples of asexual reproduction. Okay, so let's actually talk a little bit about the cell cycle. And so I'm going to draw a picture. Even though we have this picture that we can refer to quite often, I want to make sure that you have a picture of this in your notes and that you are kind of referring to that. So let's draw kind of a line here that says start. I'll move my paper up a little bit so you can see, maybe even try to focus this a little bit better. And then we're going to draw a big pie chart pretty much. Please disregard my drawing abilities. Okay, but we're going to start with a process here that is called the G1 phase. And the G1 phase takes a decent amount of time, so let's make a pretty big slice out of the pie. And this is part of what we call interphase. So during the G1 phase, which by the way, this stands for the first gap phase. So the story goes that before we had really good microscopes, when scientists were observing this concept of the cell cycle, they didn't think anything was happening during this time period because they really couldn't see into the nucleus and the chromosomes. And so as a result, they called it a gap phase, thinking that there was a gap in the cell cycle. We now know that there's some decent things happening. One thing is that the cell is growing. So cytoplasm maybe is increasing, maybe a mitochondria is replicating. They kind of replicate on their own. Same with chloroplast. But then also the cell is just doing its normal things. Like we have learned about, or we're going to learn about protein synthesis. So it's going to make proteins in the ribosomes. It's going to go through, cellular respiration and photosynthesis. I'll put PS for photosynthesis and CR for cellular respiration. It basically does all of these normal things. But one thing I want you to know, maybe something new, is that the cell becomes specialized during this part. And what we mean by that, I'm just going to draw a separate arrow down here so we can take a few more notes, is that a cell starts to learn what it wants to be when it grows up, for example. So when it's born, it's just a generic cell. And it goes through this G1 phase and it's doing all of its stuff. But then at some point in the G1 phase, it starts to receive signals that are called growth factors from neighboring cells. And those growth factors are chemicals that are telling the cell what kind of cell it needs to become. Does it need to become a heart cell, a skin cell, a red blood cell, a brain cell, a nerve cell? And so this process is called differentiation. And in differentiation, it's literally becoming different. So when I had told you that it starts off as a generic cell, another word for that, and you've probably heard this, is a stem cell. So the stem cell, or the generic cell, receives the signals or growth factors from the neighboring cells to go through this process called differentiation. And so all of that is happening in the G phase. Then we have another phase, it's a little bit smaller, but it's called the S phase. And at this point, it stands for synthesis. Synthesis means to put together. And what we're going to do is we are going to put together another copy of the DNA. So what happens here is something called DNA replication. And the goal is 2 exact copies of DNA. Sorry, I'm running out of room in there. So in the S phase, we're going to go through DNA replication. In a couple weeks, we'll actually learn more about that process. But in the end, our result, our desired result, is to have 2 identical copies of our DNA. And so that's what we call the S phase. Okay, and then there's one more phase that's going to happen. This is called the G2 phase, and as you can guess, this is the second gap phase. And again, it goes back to not really understanding or knowing what was happening during this particular phase. So we thought that there was a second gap in the cell's process. In this phase, let me circle it, the cell is preparing for mitosis. So it's getting ready. It's going to double a lot of everything. Cytoplasm is going to double. We're going to increase the number of organelles that we have. We're really just gearing up, saving some energy to go through mitosis. So I do want you to know, and I'm going to use a different color, that this whole process, I'm kind of going to zigzag. I don't know if you can tell that's a purple color. Probably should have used something a little bit different. But this whole thing that we just talked about, all of these phases, a huge chunk of the pie, all of this is what we call interphase. So all of this that we just discussed is interphase. Now this small pie slice over here is when we get to mitosis. And I know you guys have learned about mitosis in your earlier science classes, but you know that this is prophase, metaphase, anaphase, telophase, and then at the very, very end, technically not a part of mitosis, there's a little tiny step called cytokinesis. Oops, cytokinesis. And cytokinesis will be the very last step in the cell cycle. And so this is then where we end. And so let's draw a picture. We started with one cell, and we are going to end with two cells. This is called the parent cell in the beginning, and the cells that we end with are called daughter cells. We do not call them son cells because we know girls rule. Right, ladies? Okay, so let's move on to a couple of things. Hopefully you're kind of writing this out very similar to how I am on the page. That's why I'm purposely using notebook paper like this to try to estimate kind of spacing. But I want you to leave an area blank because I'm going to have you watch a video. So right here in this area, in fact, I'm going to write it in pencil so you can see different. You're going to watch a video on what we call cell cycle checkpoints. And I want you to answer a couple of questions about these cell cycle checkpoints. Number one, I want you to know, what is a checkpoint? Number two, what are the options at a checkpoint? The cell will have options at a checkpoint. And then the last thing is, let's talk about, when do the checkpoints occur? So you're going to watch this video, which I have linked for you on our learning management system on Schoology. And the video is just a couple minutes long, but you want you to listen to the narrator and watch the visual. They actually go through a visual like this cell cycle. And I want you to listen about these checkpoints. They're going to talk a little bit about that. Okay? All right, so let's continue, though, with our topic of the cell cycle. So the next thing that I want to talk about, my paper's got some splotches. Let me get a new one here. So the next thing that we want to talk about is parts of a chromosome. And I also want to make a connection for you about how a chromosome and DNA are related. So let me show you a picture that's called parts of a chromosome. And by the way, this picture is available in the PowerPoint. So this mitosis PowerPoint I am going to put on Schoology so that you can go back and take a look at the PowerPoint, look at questions, look at diagrams and answer any questions that you may have. So we're going to talk specifically about this chromosome structure here. This is a chromosome. This is actually two identical chromosomes, and I'll mention that as we learn in our heredity and DNA unit. But I want you to kind of take a look. If we were to zoom in on one of these parts of the chromosome, and if I were to actually start to unravel it, I have what's called kind of like a spiral cord here. If you've ever seen an old-fashioned telephone, and I hate to say it because those are the phones I grew up with, but old-fashioned telephones that had cords had these spiral cords like this. But that spiral is actually spiraled a couple of times. It's wound around these proteins that we call histones, and if I truly unwind it, then I get to DNA. So based on this picture, what I want you to know is that a chromosome is made of DNA. It's twisted, folded, wrapped around, smashed, wound together. We're winding it as tight as we possibly can to scrunch a whole bunch of DNA into a chromosome. But in your notes, let's actually put a little star, and we want to say that DNA is in the chromosome. I just want you to know that relationship, that the DNA is smaller than a chromosome, and we have to wind it up really, really tight in order to make it fit into a chromosome. And hopefully you already know that we can find chromosomes in the nucleus of the cell. So let's also put that as a star over here. So chromosomes, because chromosomes are really made of DNA, are found in the nucleus. Okay, I'm going to show you a picture. Again, it's in the PowerPoint if you want to go and take a look at it, but we are going to draw this structure of a chromosome in our notes. But you can see here a cell, here's the nucleus, and these little X-looking things are the chromosomes. So let's zoom out to a chromosome or zoom into a chromosome, and let's talk about the different parts of a chromosome. So the picture that you're seeing kind of looks like, almost like rabbit ears. Okay, and as I mentioned, what you really have here are two chromosomes, okay? So if I put a dotted line right through the middle of my picture, I actually have a chromosome on each side. So here is a chromosome, and here is a chromosome. What we have here is actually a duplication of the chromosome. So this is duplicated. Both of these sides are identical to one another. So we refer to this as sister chromosomes or sister chromatids. And, honestly, at this point, don't get too caught up in what's a chromosome versus sister chromosomes. We're really just going to refer to it as a chromosome. We will get into some more technical terms in about a month or so because we'll specifically spend a unit talking about DNA and the different parts and how we inherit it. But I do want you to know that this location here in the middle where the two chromosomes are connected is called the centromere. Then we have arms. So we have short arms and we have long arms. Oops, I should put the word arm. And then I also want you to know the end region is called a telomere. So like I said, this is the end region of a chromosome. If you're real interested on DNA, I just read an article about some new things that they found out about the telomere. Telomeres are indications of aging and natural deaths, people that die at old ages just from natural causes. It has to do with this telomere region starting to disappear and your chromosomes getting shorter and shorter. I just uncovered, scientists uncovered some interesting things through their research on that. Just a side note. But then also, when you see pictures of DNA, you'll see these, patterns, what look like stripes almost. I'm just putting a few. Notice that my stripe pattern is the same on both. Put a squiggly line there, thick line, thin line, however you want to do it, maybe 2 lines. They're the same on both because these are identical, but this banding pattern is usually referring to different genes. So that would be a gene, this would be a gene, this would be a gene, this would be a gene. And I want you to know that a gene is a section of DNA that codes for a particular protein. And again, we will talk more in detail about that, but this is the first time I'm going to introduce that word to you, and we are going to use it quite a bit. So really start to, encase this vocabulary into your memory, because you definitely want to make sure that you're on top of those concepts. Okay, so these are the different parts of a chromosome. Kind of underline that to highlight that a little bit. And those are important because now I'm just going to kind of walk you through the different parts of mitosis. We have already talked about interphase. We talked about everything that happens here. I just want to quickly mention what happens here in prophase, metaphase, anaphase, telophase, and cytokinesis. Not a lot of notes because I do think you have had this information before, but some things that I want to point out to make sure that you understand. So now let's actually talk about the mitosis phases. And we start with prophase. Prefix pro means to come before. Okay, so in the prophase, what we see is we see chromosomes becoming very thick. In fact, they're very thick. They're so thick, that we can start to see them underneath the microscope. What we're also noticing at this time is those centrioles. If you remember back to the organelles that we learned about, the centrioles, they're present. They actually came about in the G2 phase. In fact, I should go back and mention that, right? I'll add that to my notes. Centrioles appear in that G2 phase since we're prepping for mitosis, and that's what centrioles are there for. Those centrioles in the animal cells start to migrate, and they're going to migrate to opposite ends of the cell. So if I were to draw a picture, what I would really see in a cell, this is an animal cell, is I would see a lot of threads. It looks like a bunch of string just all tangled up. That's really all I see in my prophase. Okay, then we go into metaphase. And think of meta means middle. So at this point in metaphase, we're going to notice the chromosomes are migrating to the middle of the cell. The other thing that is starting to happen is those centrioles make these strings. They're kind of like, think of them like Spider-Man's webs, but they're called spindle fibers. And the spindle fibers are going to start to reach out because they want to start to kind of get involved with those chromosomes. Now notice when I draw it, I'm starting to elongate my cell a little bit. And now what I'm going to see is a bunch of my threads, but the threads are starting to organize themselves in the middle of the cell. Okay, then we go to anaphase, and if you think of ana as away or a, at this point now, the spindle fibers have attached to the centromere. Go back up and refer to the centromeres, that middle part of the chromosome. The spindle fibers have attached to the centromere, and they basically, it's like a fishing line. You're just going to reel them in. And so the chromosomes are pulled apart. I always think when I start to see this in the cells, it almost looks like fingers are starting to point at each other. Because we're starting to see this movement, and we're really going to start to move towards these dots. Almost think of that like the North Pole and the South Pole, and that's where those centrioles are. And so they have these almost like invisible threads that are hooked to the centromeres that are just reeling them in like you reel in a fish. Then the last phase is telophase, and think of T equals 2. We don't quite have 2 cells at this point, but we do have 2 groups of chromosomes. And these two groups of chromosomes are at the polar areas of the cell. So now we're really elongated, and now we see these threads. They're still somewhat thick, but we have two areas of them. And I'm just kind of, I guess I'm still making them look like X's. Okay, but we're starting to see that now. We have two different groups of our chromosomes. Okay, and then the very, very last step will be to make some type of division right here in the middle of that cell. And this is the process that we call cytokinesis. And cytokinesis is a little bit different between a plant cell versus an animal cell. A plant cell, remember, has that very rigid cell wall. And so as a result, it doesn't really squish. It's not going to like pinch in to really cut that in half. Instead, it's just going to grow a protein wall through there pretty much that we call a cell plate. Okay, so wall called cell plate grows. In an animal cell, we do have some squishiness. And so as a result, there's another protein. We don't call it a wall, though. We instead call it a ring. And the ring just tightens around the middle, and it finally pinches it off. This protein ring we call the cleavage furrow. And I do have some pictures of this. Again, you can check out the PowerPoint that I post. But here you can see some differences between the two. So here we have a plant cell. You can just see the beginning of that cell plate in here that's starting to grow some proteins. And here we have that cleavage furrow, the pinching that's happening on an animal cell. Just a couple other things that I wanted to show you and talk a little bit about is there are certain organisms that we use in biology classes to teach students about mitosis and to be able to identify cells in various phases. And so what we typically look at under a microscope is the very end of a root. So think of this as being underground, and this is the end of the root. This happens to be from an onion plant. Seems like we use a lot of onions in biology class, don't we? But a couple of things to point out. If you remember, one of the reasons why we do mitosis is to grow. And so this is actually where a plant grows, is right here at the end. It doesn't grow up here, it's growing down here. So this is the area that we expect to see a lot of mitosis happening. If I were to keep looking on this root all the way up in my microscope and maybe look at an area somewhere up here, I would no longer see any cells going through division. Remember, division is mitosis. But interphase is the main part of the cell cycle, just the everyday life of a cell. I would see a ton of cells in interphase up here because that section of the root is no longer growing. It's here at the end region that we see mitotic phases underneath the microscope. And if I were to zoom in really, really close to those, this is kind of what I would see. And there's a couple things that I wanted to point out because you do need to be able to identify what these phases look like. Most of the phases that you see just look like somewhat dark circles. Those are all examples of interphase. You can see how many. There's a bunch of interphase. But sometimes those dark circles start to get splotchy, and then they start to get stringy. When they start to get stringy, then we know we are looking at prophase. That is the beginning of mitosis. Chromosomes get really, really thick so that we can see them. After prophase comes metaphase, where they line up in the middle. So when you see pictures of cells and it looks like there's a dark line in the middle, then you know you have metaphase. After metaphase comes anaphase, where things are being pulled away from each other. And so you can see right here, we're starting to get two congregations of chromosomes, and they're being pulled away from each other. This is a great picture of anaphase. And then at the end, we have two, or telophase. And you can see here, there's no division in that cell. So it's still just one big cell. You can kind of see the outline of it. But we have two congregations of those chromosomes. And so that's what we call telophase. I'm going to run out of room here. And that's telophase. So you'll be working on practicing identifying these. These are common questions that I have on quizzes or tests, being able to identify the general phases. And then, like I said, when you see these ones that are just the dark blobs, those are interphase. And it shouldn't be alarming to you that we see a lot of cells in this interphase. Because if I go back to our very first picture that I showed you, this whole part right here is interphase, all of this. So we expect to see a lot of cells in those. It's not till we get to the prophase, metaphase, anaphase, telophase, a little bit of cytokinesis, that we have smaller and fewer cells in those particular phases. Don't forget, after you've watched this PowerPoint and you've taken, or this video and you've taken notes, please go to the cell cycle checkpoint video. Make sure you watch it and answer those questions to fill in into your notes. And we'll talk about those questions in class, but you have to watch the video first, please.